there are a number of interesting seminars in addition to today's, which is of course the most interesting of the week, but there are others this week. Later today, PEI is co-sponsoring with our colleagues in the Center for Human Values, a talk by David Antoff from Berkeley on inequality and the social cost of carbon. Tomorrow we have an environmental humanities talk in the afternoon for those who have been following that series. And there's also a special lecture tomorrow for EEB called the Frontiers in Biology lecture given by Kathy Willis from Oxford. The title of Green Health, the science behind why nature is good for us. That's a very good message. And for those who are interested in a late Friday afternoon seminar, 4:30 on Friday, another really interesting talk on the politics of water in China, jointly sponsored with us and the Center for Contemporary China on campus. This is by Scott Moore from the University of Pennsylvania. So a few things going on this week. But we get on to today's lecture, which of course is by Bess Ward. Bess is the William J. Sinclair Professor of Geosciences. Also has part of her appointment in PEI, so she's one of our core PEI faculty members. And she's also the department chair for Geosciences, position she's held since 2006. Bess has been active in PEI in many ways. Some years ago, she taught one of the introductory courses, ENB 202. I remember it well because at the time I was teaching 201 and she always did a better job in 202 than I did in 201 and got more students. And eventually I quit. And if you noticed this, there was a very nice write-up on a course that she taught last spring here at Princeton. And the interesting part of it was that for 5 weeks she was at sea taking measurements and figured out how to continue teaching her course while out doing field work in a way that I thought was quite fantastic, and you can find it on the university homepage and other places. She was out at sea because her focus is largely on oceans, and interest broadly in the nitrogen cycle, particularly the marine nitrogen cycle, with a focus on marine bacteria and the kinds of processes that these bacteria tend to have, and it's consistent with, of course, the title of the talk, which will be focused specifically on N2O, of course, a piece of the nitrogen cycle. Bess has received many awards. I'll just mention a few. She's a fellow of the American Academy of Microbiology. She's a fellow of the AGU. She's a fellow of the American Academy of Arts and Sciences. And a few of her most recent awards, she's received the Rachel Carson Award from the AGU. She was the Charnock Lecturer at the Southampton Oceanography Center in the UK, and the Marie Tharp Award from the Helmholtz Center for Ocean Research in Germany, among a long list of others. Please join me in welcoming our good friend, Bess.
Sound check, is that working?
Yes.
Okay, great. Thank you, Mike. Just to set the record straight, when I was first hired as part of PEI and Geo, the first thing that they did was sit Mike and me both down and say, okay, fix these courses. And so we spent hours and hours every week, meeting every week, to revamp those courses. And then we thought 201. We taught 201 first, and I went to every one of Mike's lectures, and that's how I learned to do 202, which we did the next semester. And I'd also like to acknowledge, while we're talking about those courses, the incredible contributions of Henry Horn, who was, I believe, the founding director of PEI and who recently died. He spent that summer with us, too. And it was his incredible generosity and humor and insight that really helped us both immensely develop those two courses, which we did for about 10 years, and then we're glad to turn them over to the current people. But it was that experience teaching 202 and in the ENV program and the sort of global view of PEI that really broadened my perspective, and you'll see, I hope, a little bit of that in this focus on nitrous oxide, which is a really interesting component of the nitrogen cycle and the microbes that do all this fascinating stuff with nitrogen, but it also has global implications. Nitrous oxide was discovered in 1788 by Joseph Priestley, great chemist. And it had two important uses in humankind. And it's still used in both of these ways. One is as a party drug. So this was in 1820. And there's the little balloon of nitrous oxide. And apparently it brought a lot of joy to a lot of people. That's why it's called laughing gas. It makes you laugh. I'm not going to tell you any of my personal experiences with it. And its other use was as an anesthetic, right, for dental surgery in particular. You come out of the dentist hurting like hell, but feeling very happy. And it was about 200 years later, after its discovery, that Ramanathan at Scripps Institution of Oceanography realized that trace gases, including nitrous oxide and methane, could actually be, although they're very, very trace level components of the atmosphere, could add detectably to the total radiative forcing and therefore global warming of the atmosphere. And he predicted that it would be about 2010 when we would actually be able to detect that influence of the trace gases. So in 2009, that's why when it was recognized very forcefully in this important paper that nitrous oxide is no longer a laughing matter, because it has now become the most important ozone depleting chemical. We've sort of got CFCs, not completely, but for the most part under control. They were extremely potent and all man-made. Nitrous oxide is mostly not man-made, and it's a powerful ozone depleting chemical with the chemistry that happens in the stratosphere. It also has some global warming potential, which is increasing, partly because its concentration is increasing, but molecule for molecule, it has about 300 times the global warming radiative forcing of a molecule of CO2. So even though it's a really trace level, its global warming potential is increasing and not in competition with CO2 or methane. But you look at the size of these wedges, the uncertainty is large. So the actual amount of forcing and ozone depletion due to N2O is going to be large and increasingly important, but incredibly uncertain. And as I mentioned, it's important to note that N2O has a natural source. Most of its source is natural. And this paper is just one of many that you can find that will divide out the sources of nitrous oxide. And you see that this is the total, and this is the anthropogenic part, this pink wedge. So about a third of it is anthropogenic, and that's mostly agriculture and fossil fuels. About 20% of the total is distributed to the ocean, and the rest of it to terrestrial systems, not counting agriculture. So that's the sort of big picture is where nitrous oxide comes from. And I think a lot of people agree on sort of the proportions here, but I want to give you an idea of the uncertainty in the total numbers, which is what drives our interest in trying to figure out what controls the production and consumption of nitrous oxide, because there's an incredible amount of uncertainty and disagreement in the literature over just the total flux of nitrous oxide to the atmosphere. I could have picked a lot more references to confuse you further, but this is enough to get the point. That was Davidson and Cantor, the one that I just showed you in the pie chart. Their natural part, not even all of it, their part that they attributed to being natural was on the order of 10 to 12 teragrams of nitrogen per year. The IPCC, generally taken as the global authoritative number on this, says that their total, including anthropogenic, is only on the order of 6, but it has a huge uncertainty. These guys have been modeling it for a while, and this is one paper they have two estimates of just the ocean part, and you see that that could be as much as almost the total, which everybody thinks is only 20% of the total. And another arrived at with a completely different kind of calculation, was a bit smaller. That part of that model was what we used in this one after we reparameterized some of the processes that I'll be telling you about, and then we come up with a number that's somewhere in between. But if you look at where all those arrows are, we don't know. There's a lot of uncertainty. The reason that nitrous oxide is of interest in the ocean is not just that 20% number, which you can take with some uncertainty, but the distribution of nitrous oxide in the ocean implies that it could be a larger source to the atmosphere than we think it currently is. And this is the distribution with depth. This is I think of there being sort of two graphical presentations that are the oceanographer's view of the world, and this is one of them, concentration on the x-axis and z, the depth axis. So concentration or rate or something, and then everything with z, with depth. So the distribution of nitrous oxide with depth in these particular parts of the ocean called oxygen minimum zones, OMZs, is where there is the most nitrous oxide in the water, and there's this big peak near the surface of the ocean, close enough that there's a diffusive flux out, and close enough that it could easily shoal up next to the coast and burp out in large chunks. And just to give you how big a number is that, nanomolar doesn't sound like very much, but if it was just in equilibrium with the atmosphere, it would be on the order of 10 or 11. So it's tenfold saturated, so something is happening here, both production and consumption most likely, and that's what I'm going to talk about. And to do that, I'm going to talk first about the nitrogen cycle, but before that I want to acknowledge Amal Kumar is my most important collaborator here at Princeton, and he's the king of N2O. See, that's the N2O. Amal tends to celebrate his birthday at sea. I don't know why, we're always at sea in April, and so that was his birthday cake. And I thank him for all of his work on both N2O and DNA, that's his other metal. Okay, so we're going to talk about nitrous oxide in the ocean. To get there, we're going to have to understand something about the processes that both produce and consume nitrous oxide, and that'll help us figure out why oxygen minimum zones are so important as hotspots of potential and real nitrous oxide emission to the atmosphere. And to get there, we have to understand the nitrogen cycle. You know I couldn't get away without talking about the nitrogen cycle in some detail. And that's going to involve microbes and the microbial communities and assemblages that do all these processes. And then we'll wind up thinking about what does that mean for nitrous oxide in the future. And so to understand the nitrogen cycle in the ocean, I want to start out at the very beginning with the question, why is the ocean blue? And that has something to do with the light penetration and the spectrum of the electromagnetic spectrum and so forth. But it's really blue because there's nothing in there to absorb the light. So this is a picture I took in the Sargasso Sea. Yes, that was a view from a research vessel. That's one of the cool things about going to sea. If there had been anything really in the water, phytoplankton, plants of the ocean, it would look more like this. And so the reason that that involves nitrogen is that there's nothing in the ocean, in most of the ocean is blue because there's not enough nutrients to support phytoplankton like this. And this takes us to Liebig's law of the minimum, which explains for plants, in this case phytoplankton, that plants will only grow to the extent allowed by the least abundant resource, and that's in proportion to the ratios in which they need all those elements to build biomass. And so this is obviously a freshwater informed bucket, but in this case, the nitrogen is the lowest slat of the barrel, and you can only accumulate water or biomass in proportion to how much nitrogen there is if that's the least present, least abundant in proportion to how it's needed in the biomass. So it's been shown many times, and it's generally accepted that lakes and ponds are generally phosphorus limited, but the ocean is generally nitrogen limited. And the implication of that is that if it's limited, and that's what's constraining the amount of water in the bucket or the amount of biomass, is that if you add more nitrogen, then you'll get more growth. So you add more nitrogen, you'll get more phytoplankton, and then you'll get more fish, and that's what controls the productivity of the system. So that's the scientist in you will be saying, well, let's do the experiment. Let's see if that works. Well, they did that in lakes in Canada. They're blessed with a whole bunch of little lakes, and they number them. The highest number I think we saw was 224, but there's thousands of lakes. In this particular experiment, this was a lake that was conveniently divided with a movable barrier between a narrow point in the lake. And to one part they added both carbon substrates and nitrogen, and to the other one carbon, nitrogen plus phosphorus. And in this lake, sure enough, phosphorus is limiting because you add more phosphorus, you get more stuff. So a lot of growth of no longer blue water because it's full of phytoplankton. So what that would mean was that if you were to be able to do the experiment in the ocean, and Wally Broker famously said, at least to me, I remember it from a session that was all about those lakes in Canada. He said, well, I work in lake number 1, and that's the ocean. And it's really hard to do the experiment in lake number 1. However, we are doing that now, as we'll come back to. But the implication is that if nitrogen is limiting, lack of nitrogen is what keeps the ocean blue for the most part. And in fact, lack of nitrogen is easy to believe. This is the other oceanographer's view of the world, which is a map showing distribution of something on a color gradient. And this is the distribution of nitrate in the surface ocean at the time when it is maximum at every place, and at every place when it is minimum. And the point is that it's all purple. So most of the surface ocean most of the time has no nitrate. And that's one of the very popular forms of nitrogen that phytoplankton use for growth. And so over vast expanses of the world ocean, nitrogen is in fact limiting, meaning that where you have more nitrogen, you should have more productivity. And that we do find to be the case. This is not exactly productivity, it's chlorophyll from satellites. But you see the highest chlorophyll regions also do correlate with highest productivity, and they tend to be where there's deep mixing seasonally or where there's upwelling. And those regions are either nitrogen and or iron limited. Sorry. So what happens in the surface ocean that drives everything else that happens in the nitrogen cycle is the production of organic matter from those phytoplankton that are supported by nitrogen being upwelled in those regions. And most of the productivity, all of the primary productivity in terms of photosynthesis happens in the surface ocean. This is a very, very simple representation of that it produces oxygen and produces organic matter. And also, most of the utilization of that organic matter, most of the consumption of it by heterotrophs, so plankton, fish, and everything, also happens very close to the surface where the food is being produced. And that then consumes oxygen and consumes that organic matter and takes it back to CO2 and releases the energy that everything needs to grow. So this cycle happens really rapidly and recycles just about everything in the surface, but some things escape and fall out. And so there's a measurable vertical flux. Not this dense, this is way overestimated, the density of stuff in the water. But a little bit of it sinks and can be permanently buried. A very small amount of it actually makes it to the bottom. But if you want to measure that, that's the stuff that's actually going to support everything that's happening below the surface layer. It all depends on the surface and it supports everything that happens below that. So oceanographers measure that flux by using a variety of styles of buckets. So each of these is just a different kind of bucket that you put out there and catch what fell into it. And this very famous plot, which is a summary of the Vertex program by John Martin et al., this is where they put the buckets and that's how much stuff they caught. And so you see that nearer the surface, you catch a lot more. And as you get down to 1,000 meters, there's not much. You can measure the bottom of the ocean is down here at 4,000. It's really, really not much. And so what that tells you is that the stuff that went missing between here and here, for example, was consumed in the water column. So most of it is recycled up here, but none of it escapes recycling. And it's used up and consumed by microbes and other organisms so that less and less of it actually escapes to sink all the way to the bottom. And you remember my little equation for photosynthesis and organic matter cycling. That means that oxygen is being consumed during this process. And if you have a lot of organic matter flux and not very much ventilation, then you use up all the oxygen. And this is an actual measurement from ETNP. And that produces what we call an oxygen depleted zone. We now know, I'm not going to go through the story of how we really know that now, but it's an instrument challenge, that there's essentially zero oxygen between 100 meters and 500 or 600 meters. So there's this really thick zone in the water column that has essentially zero oxygen. And so that is because organic matter has supplied the utilization, the respiration of the organic matter and the use of oxygen. And now they've run out of oxygen, they have to do something else. And that is how the oxygen minimum zones come about, the consumption of organic matter and the consumption of oxygen faster than it can be replenished by the circulation of the ocean. And how does that connect to the nitrogen cycle? Well, when you run out of oxygen for respiration, you have to use something else, and the next best thing is nitrogen. And in order to understand why that matters, I have to tell you about about the nitrogen cycle and how we distinguish between fixed nitrogen and not fixed nitrogen. So most of the nitrogen in the atmosphere and in the ocean is N2, and obviously that's not a good substrate or we'd all be eating the air, right? Most organisms cannot utilize N2. A very few of them can, and they produce fixed nitrogen. So fixed nitrogen is everything doesn't have a double or triple bond between the two N compounds. And that's not very much. It's organic matter, nitrate, and ammonium. And if I were to put these two to scale, you couldn't see this dot. All right, so most of the nitrogen in the world is not fixed and can't be used by most organisms. So that really puts a premium on those who can. And those are the nitrogen fixers. And they're all bacteria and archaea. And not very many different kinds compared to the total diversity of microbes in the world. But they affect a little bit of a flux from this huge pool into the fixed nitrogen pool. And so what really matters to organisms and the availability of nitrogen when we talk about nitrogen limitation is the fixed nitrogen pool. And that pool is decreased by two processes, also microbial, that affect nitrogen loss, as we'll call it, and that's denitrification and anammox. And they both take the fixed nitrogen and turn it back into N2. So these two processes control are all controlled by microbes, and they all control the fixed nitrogen inventory. So that's what is going to set the amount of nitrogen that's available to support primary productivity. And the connection with oxygen is that both denitrification and anammox are strictly anaerobic. They only occur in the absence of oxygen. And so that means that places where they occur in the ocean is going to be those places where we had very little oxygen. So that's the 0, pink. And those are the places where, by this measure, N star, which is, if it's negative, means that some of the fixed nitrogen has gone missing. And that occurs basically in 3 places in the water column of the world ocean. It also occurs widely in sediments. But these are the places where we've focused our attention on the nitrogen cycle of oxygen minimum zones, because that's where fixed nitrogen loss occurs, and that's where nitrous oxide turns out to be a really important part of the nitrogen cycle of the ocean. So to summarize the motivation, nitrogen limits primary production in vast regions of the world ocean. When nitrogen is available and organic matter is produced in the surface, then it sinks, at least some part of it. It's decomposed at depth, and that leads to depletion of the oxygen underlying those highly productive waters. Fixed nitrogen then is removed during the decomposition of that organic matter by microbes in anoxic conditions in water and sediments. And that means that the microbial biogeochemistry of low oxygen environments controls the nitrogen inventory of the ocean. So that's why we care about the processes that happen in low oxygen. So oxygen minimum zones, or the regions of the ocean where these processes occur, are characterized by these very typical distributions of things. This is the ODZ we saw with the oxygen, so the part of the water column where there is no oxygen. Typically, you see 2 nitrite maxima, one little one up here called the primary and one called the secondary nitrite maximum. This comes in various shapes and sizes, but it's always below the limit where there's no oxygen. In fact, nitrite is a really good indicator that there's no oxygen in the water column. The other thing that is characteristic in terms of nitrogen distributions in these systems is this big nitrous oxide maximum. And so we just saw this, I introduced this originally with this big maximum, which is right in the gradient of the oxygen profile. And so those things, whenever you see a maxima like this, you know that something is happening to maintain it, because diffusion would ruin it, would take that peak away rapidly. So there has to be production, and there would be loss by diffusion, but also probably production and consumption by microbes. And so what I'm going to do is walk us down the water column, looking at the microbial processes that occur in these relative intervals. And I'm not going to don't hold me to the actual depth interval, but they're in this sort of order, from the surface all the way down into the deep ocean, and here's the oxygen minimum zone. And here are this slide will appear several times, so don't memorize it yet. The test will be at the end. So these are the key processes that occur in each of those levels, the kinds of microbes that are essential to the key processes and what they do for a living. So we'll start in the surface. That's where we just talked about primary productivity being important, being the main thing, and that drives everything else below it. Those are our photosynthesizers, and the ones that are really important in the world's most productive ecosystems, everyone thinks of as diatoms. So they're big, fast-growing cells. They love nitrate. They do really well on nitrate, and they feed and support the world's shortest and most productive food chains. But there are a lot of other things that are also important in primary productivity. Some of them turn out to be really interesting and somewhat peculiar in oxygen minimum zones. So we look at the distribution of phytoplankton. It's easy to do by measuring the fluorescence, which represents the chlorophyll. And so this is a trace from that cruise actually last year when I was playing hooky from teaching biological oceanography. And this is the distribution of chlorophyll with depth. So there's a subsurface chlorophyll maximum, and there's a deeper one too. And this is the one I want you to look at. This is where we can imagine diatoms being, and lots of other phytoplankton. But this is the oxygen profile from that cast. And you can see that it goes pretty much to 0, somewhere between 100 and 150 meters, and there's a secondary chlorophyll maximum clearly in the anoxic water. So these are weird little phytoplankton. It's almost dark down there too, right? The light profile basically goes like that. So it's almost dark and it's anoxic. So who are those guys? Well, with the help of, we didn't figure this out, right? We knew this before, but with the help of Ashley Maloney, who was on this cruise, we were able to collect Bob? Just turn that thing off. we were able to collect cells from those discrete depths where the chlorophyll maxima were, and to target them with a particular size fraction. So we're looking at the size, the microbes, the cells that are present in the water column that are between 2 microns and 0.4 microns in size. So tiny little guys, clearly not diatoms. And the ones that were most important in that size fraction in this chlorophyll maximum are these red things, which are Synechococcus, which is a cyanobacterium. And the ones that are important here, this is like what you get when you filter 400 liters of seawater. Those are the little guys. There's a lot of them. These are called Prochlorococcus, and they're another cyanobacterium, and they're the ones who are stupid enough to live in this anoxic, dark seawater. And it's really hard to understand how that happens, but I remind you that this peak is maintained. So they have to be doing something, even if it's not much. So Emilio Garcia Robledo et al. Had investigated this on several past cruises, and they showed that there was indeed gross carbon production and oxygen and carbon fixation and oxygen production at very low light levels. Now the actual light level where they collected the samples was somewhere between 0 and 10, but they just couldn't control the light to get it lower than that. But it proves, even if we don't know the exact rate at which they were doing photosynthesis in situ, that they are capable of doing photosynthesis, and they're doing it there. However, remember, this is where we could not measure any oxygen. So they proposed a cryptic oxygen cycle, meaning that oxygen is being produced and consumed, but you never are able to measure it there. So there's another process going on that's consuming it, and of course that could be heterotrophic respiration at really, really high affinity for oxygen. But the part of the nitrogen cycle that's interesting there, now looking in this region, the upper oxycline and the top of the ODZ, where the photosynthesis by Prochlorococcus is important, is nitrification. And nitrification, we've just run out of charismatic organisms. Everything else is going to look like a little round or oblong jobby, because bacteria just aren't that charismatic, at least morphologically. So these are ammonia oxidizing archaea. They do the first step of nitrification, which is ammonium oxidation to nitrite. And then the second step, nitrite to nitrate. So this regenerates our most popular nitrogen nutrient form, is performed by nitrite oxidizing bacteria. Most important one in the ocean is Nitrospina, we now know. And so the odd thing about this process, it's not odd, it's odd in the context of where we find it happening, is that they both require oxygen as far as we know. And ammonia oxidizers require oxygen as the very first enzyme, as a monooxygenase, and they incorporate dioxygen into the nitrite. Nitrite oxidizers require, because they're oxygen respirers, so it doesn't actually take the oxygen from O2 and put it into nitrite, but the metabolism of the cells still require oxygen. But they could be the ones who are utilizing the oxygen that Prochlorococcus is producing, because when we measure their rate distributions, here's the distribution with depth again of ammonia oxidation in the East-tropical North Pacific, and it has a maximum right at the top of the ODZ. Also similar in the ETSP, Eastern Tropical South Pacific, oxygen minimum zone. And nitrite oxidation shows a really high rate, much higher than ammonia oxidation, and it's below the oxycline. It's where there is no oxygen. And this is something, the first time we published it, everybody went, no, just don't believe that, must be something wrong. So we've done that several times, we always find it, and we're trying to figure that out. And how that works, because it's so widely understood and accepted that nitrite oxidation requires oxygen. So the person working on that in my lab now is Shin Sun. And I know this was a cruise in the tropics, but we had to do some of the work in the cold room because the seawater is cold. And so you're doing a lot of manipulations. And this is what she thinks apparently about working on an anaerobic process that's not supposed to be happening. But she's made some really incredible findings, which are really cool, based on metagenomics. And I don't have time to go through this in any detail, but what you do is you filter out the DNA that's in seawater, sequence it, put it back together again to figure out who it came from, even if you've never met that organism before. So it's really cool. They're called MAGs, metagenome assembled genomes, and NOB is nitrite oxidizing bacteria. And so she was actually able to find what looked like 2 new species of nitrite oxidizing bacteria defined by their almost complete genomes, and to estimate their relative abundance, and this is the ODZ again in gray, And you can see that both of these forms look a lot like the distribution of nitrite oxidizing activity. So there's not a whole lot in the surface. There's a maximum somewhere near the top of the ODZ, and they're both present in the deep anoxic water of the ODZ. So there's another, so we have the rate measurement and we have the distribution of the NOBs, and it looks like nitrite oxidation should be happening there, even though it shouldn't be, because it's anoxic. Another piece of evidence is derived from Karen Cassiotti's group, who's on the same cruises, who developed models based on the natural abundance of stable isotopes of nitrogen and oxygen. And they find that their model, based on just the standing stock concentrations of the isotopes, requires that you have nitrite oxidation going on in the anoxic layer right there at about the rate that we measured it happening. So that's like 250, 300. That was the number. So at a pretty high rate. But the same model predicts that you have to have nitrate reduction going on because you're not accumulating a lot of extra nitrate and you're not consuming all the nitrite. So you have a nitrate and nitrate reduction and nitrite oxidation going on at relatively high rates just there at the top of the ODZ. So who's doing the nitrate reduction? It turns out that that is the most popular organism in the ocean, SAR11, and that's known as a small metabolically restricted, stripped down genome kind of thing, SAR11, but it's known as an aerobe, but it possesses nitrate reductase genes. And these are all the different clades that have all contain nitrate reductase, and you can see that this is another depth profile. They're very abundant. They're up to 30-40% of the total assemblage, and they're capable of nitrate reduction. So it looks like, this is what SAR11 looks like. It's tiny, but it's ubiquitous. So it looks like it's the one doing the nitrate to nitrite, and that we have this sort of feudal cycle of nitrate being reduced to nitrite and nitrite being oxidized to nitrate. And there are a lot of things about this that we don't understand. But it does play in further to the nitrogen cycle of deeper layers in just a moment. So this is something that we don't understand how this happens in the absence of oxygen. But this we usually think of as just the first step of denitrification, the complete reduction of nitrogen oxides to N2 gas. So denitrifiers, so that's, now we're going into including the core of the ODZ, where both denitrification and anammox, those two anaerobic nitrogen loss processes, both occur. And denitrifying bacteria, although they're incredibly diverse, many different kinds, they're clearly not all that photogenic either. It's really misleading to look at all these pseudomonad type looking cells and understand that there's incredible diversity of them. They're mostly heterotrophs. They mostly respire nitrous oxides instead of oxygen, so they're taking care of the organic matter. They're decomposing that even when organic matter is gone. The first step that we saw was nitrate reduction to nitrite. The whole process goes all the way to N2, but denitrifying bacteria come in all sorts of metabolisms, and some of them start at nitrite and go to N2, some of them start at N2O and go to N2, some of them start here and go to N2O. So it's sort of modular, so that they can, the pathway is sort of modularized and different parts of it appear in different kinds of organisms, not all one bug going straight from nitrate to nitrogen gas. But importantly, these guys do require organic carbon, so they're heterotrophs. They're degrading organic carbon. They're producing ammonium, remineralizing the nitrogen, and consuming nitrate to make N2. The other microbes that are involved in nitrogen loss processes are anaerobic ammonia oxidizing, or anammox bacteria. And they use the nitrite and ammonium that is produced by denitrifying bacteria, either as intermediates or as the decomposition of organic matter back to ammonium, to make more N2. So these are the two processes. And the anammox bacteria are autotrophs. They fix their own CO2. It's really a hard way to make a living because there's not a whole lot of energy to be got from this process. They actually get their energy from the oxidation of nitrite to nitrate, which is really a poor way to make a living. So these two things, extremely different metabolisms, actually depend upon each other because these guys require the are the ones who process the organic matter and produce some nitrite and make ammonium. These guys require the production of those two things thanks to denitrification. And there was a bit of a controversy over how much of each of these produced contributed to the total loss of fixed nitrogen, and we hypothesized that that had to depend on the organic matter. So everything depends on the organic matter that starts at the surface of the ocean. And this was a thesis work of Andrew Babin, demonstrating one of his major skills, which is working in a glove bag. So these processes are anoxic, right? Anaerobic. You have to handle the samples very carefully, and we spent a lot of time trying to optimize that. So he's taking the water from the ocean and transferring it into all these little vials in which we're going to do the incubation, and then we flush thousands and thousands of these vials. I think I did about 5,000 on that cruise, and do all the manipulations in the lack of oxygen in order to estimate the rate of the processes and the ratio of them. So to summarize those experiments, we added different amounts of organic matter and measured the rate of denitrification and anammox in those little vials. And the first thing you see is you add organic matter, you get more rate. So that's back to our first principle that nitrogen limits what's going on. And that would imply in the oxygen minimum zone that the supply of oxygen from above was what limits the process. So in the control, it received only the ammonium tracer, or nitrite tracer, and these others received a little bit of extra organic matter in the form of sugar, which literally came from the galley because we didn't think of that experiment until we got on the ship. Cas amino acids, we had brought those with us, and we knew the composition of them. Particulate organic matter we caught that day in a sediment trap, right? One of those buckets. We caught the organic matter that day and fed it back to them. And we're going to ignore this for the moment. And what you see is if you add organic matter, you get more denitrification, and except for sucrose, you get more anammox. And if you calculate, based on the stoichiometry of the organic matter, how much nitrite and ammonium should be released for the anammox to use, you get an expected anammox, which is gray, compared to the measured anammox, which is black, and which our peer reviewers called remarkable agreement, which was really neat. So the predicted fraction of anammox really depends on what they're having for lunch today. And that's where this was kind of interesting. This one was actually the particulate organic matter from the ocean was not a lot more nitrogen in the amino acids. You'd think amino acids, C-to-N ratio of 5 or something, a lot of nitrogen would be a great substrate. Well, they really preferred what they were going to have for lunch that day, even though it had a lower nitrogen content. So they're utilizing the stuff that was immediately there as labile organic nitrogen. So the other part of that story, of course, is where, and the dependence, where this happens in the ocean, and the dependence of the process on that flux of organic matter from above. And that's the original Martin curve that I showed you, and this is just the distribution of the measured rates from that particular study. We've measured this a lot, many more times than that, but it generally follows a curve that can be modeled by the Martin curve formula. And this is the ratio actually had been measured in this area and converted to nitrogen. So that's what we actually measured, and that's an old measurement of the rate distribution. So everything depends on the surface, and it makes total sense that the anammox and denitrification together reflect the mineralization pattern. So back to how does our model deal with denitrification and anammox? Pretty well. It also simulated the denitrification and anammox distributions, which they have a little blip down here that we don't see, but maximum just near the top of the ODZ. And the rates are very similar to the ones that we actually measured, but they're a lot lower than the rates of nitrite oxidation and nitrate reduction. And so Andrew formalized this, or cartoonized this, in this illustration in which we have a sort of a rapidly spinning nitrate, and a little bit of it is actually completely denitrified then into gas, and some of the ammonium that's liberated in the organic decomposition that accompanies nitrate respiration is used for anammox. And so that finally takes us back to N2O, which is the final step in that sequential reduction of nitrogen oxides. And this is everything before here is fixed nitrogen. This is not fixed nitrogen. And so that's a loss of fixed nitrogen, also production of this important gas. And the person whose work I'm going to highlight here is Jimmy G., shown collecting his samples on one of those cruises, who investigated the actual microbial processes which produce nitrous oxide. And I haven't mentioned one of them before, but this is again, just the depth distribution of stuff. So that's the oxygen minimum zone. And here's the nitrous oxide peak, and here's the nitrite peak. So it's a classic, typical OMZ kind of distribution of stuff. And nitrous oxide is generally understood to be produced by both nitrification, so from ammonium to nitrite, and somehow or other some nitrous oxide is produced, and that's highly controversial right now, actually, the pathway by which that happens. But also from denitrification, because it's in that sequence of nitrogen oxides. And so what he found, and this is just one example, was that the maximum rate of nitrous oxide production via nitrification occurred just above the oxygen minimum zone, just above where oxygen went to zero, and you can't detect it in the ODZ itself. And that's sort of like the nitrification profile that I showed you earlier. Denitrification, on the other hand, so the nitrous oxide coming from nitrite and nitrate, here's nitrate, both are maximum just at the top of or within the anoxic layer. So that makes sense. We think of denitrifiers as being anaerobic. The other thing to point out, though, is that everyone considers nitrification to be the main source of nitrous oxide because it occurs all over in the global ocean, but the rates here are really puny compared to the rates that we measure for denitrification, so like a factor of 100 more you can get from denitrification. And that's because here we have this anoxic layer and denitrification is happening. Denitrification doesn't happen over the rest of the ocean, right? Only around the OMZs. So these are really reproducible. We observe these wherever we measure in ODZs. That this is the sort of distribution of nitrous oxide and the distribution of rates. But that has not informed us as much as we would like to figure out this uncertainty in the overall rates and fluxes. So I'd like to put that back together now and say, is there some way that we can inform the emissions calculations and the models based on our investigations of the physiology and the actual rate processes in the ocean? And as I said, we are actually doing this experiment in the ocean, not quite this dramatically, but we're in the middle of a huge global change experiment, and the likely changes that are ongoing now and that will continue include nitrogen deposition. So that's both from the atmosphere and from fertilizer on land. So those are the sort of net inputs into the ocean. And there's a lot more of that going on than in the past because the root of all evil is population. Population's been increasing. Global warming due to CO2 is warming the atmosphere and the ocean and leading to acidification as well. And global warming of the surface ocean also increases the stratification or the stability of the water column of the ocean. And I'd like to tell you a little bit about a model. And again, there are many that I could have chosen, but this one is fairly straightforward in understanding both the model premise and what they found. And they considered nitrogen deposition and warming both independently and together. And their specific question was, what effect will that have on nitrous oxide emissions to the atmosphere in the near future? So I bet these are too faint to be seen, but they plot between 1850 and 2100, approximately 2100, the sea surface temperature and the stability of the Brunt-Weisela frequency, which is the stability. And what they find is that the ocean gets warmer and more stable, which means it's harder to mix the surface layer with the deeper layers. The mixed layer, the part that is interacting with the atmosphere and being mixed and incorporating the oxygen from the atmosphere, gets shallower. So you have a smaller part of the ocean that can contain that oxygen. And the total oceanic oxygen inventory therefore decreases because it continues to be consumed in the deep water and there's less exchange between the surface and deep water. There's no reason to think that nitrogen deposition should have had an effect on any of these things, so surprise, surprise, warming has a much greater effect than in deposition. But that was true for all of the things that they looked at. So you could just look at warming because it constrains the mixing, basically. So the effect of of constraining the mixed layer and decreasing oxygen inventory was actually to increase the rate of denitrification, except that the overall effect is to decrease the rate of primary productivity, because with more stability, less mixing, there's less nutrients being advected or upwelled or mixed into the surface layer. So overall primary productivity actually goes down. And the stability then leads to the nitrous oxide inventory of the ocean actually increasing, but that's not because there's more being produced, it's because there's less being vented to the atmosphere. And the actual nitrous oxide emissions are forecast to decrease because it all links to primary productivity and the stability being increased, and therefore a lot less able to mix with the atmosphere, a lot less nitrogen fueling, less primary production fueling the nitrogen cycling happening below, which is where the nitrous oxide is produced. And so this is all internally consistent with how we understand the ocean works, the physics and biology. But my question was, is there anything that this model points out that we don't know, and other models that are basically like it, that we could inform by some of the kinds of measurements we can make? And I think there are two problems with it. The physiology of the microbes is not taken into account very well in the models because, as I said, they mainly consider only nitrification. The hotspots that are oxygen minimum zones, which is where we have that tenfold supersaturation or more of nitrous oxide, and where denitrification happens, are not well represented at all because you only have one function, which is oxygen sensitive. And so what we need to know is the oxygen sensitivity of those processes a little bit better. Models in general don't work very well in the coastal system either, and those are extremely variable in where a lot of the extreme emissions occur. So I have a little bit to say on how to approach those. Again, this is Jimmy's work. A lot of the model system that I just mentioned use a formulation of the dependence of nitrous oxide production from nitrification from Garreau et al. In 1980 using a culture of ammonia oxidizing bacteria. We now know that ammonia oxidizing bacteria are not important in the ocean, and when you actually measure the dependency of nitrous oxide yield on oxygen, it does increase at low oxygen, but not until you get to really, really low oxygen, which implies that denitrification is potentially a much more important source. And when you look at the dependence of nitrous oxide production from all three of those sources, you see again they all increase with decreasing oxygen concentration, but that peculiarly enough, we actually still see some nitrous oxide production in the presence of oxygen. This is increasing oxygen concentration from nitrate. And again, the rates of production are much higher from the denitrification, the reductive processes than from ammonia oxidation. So it looks like we may have a nitrous oxide production in oxic water, which is something we're still working on, but also that we need to reformulate the parameterizations of nitrous oxide production in those models. And this is what we tried to do in this model from Timmy's paper. And I just want to point out that the hotspots really are where we said they were in the oxygen minimum zones, and that those are disproportionately important over the surface of the ocean, such that denitrification is responsible for about half the total in the ocean. And so why is that? What is that effect is that going to have on the efflux? This is to my point about the coastal region being really important. These were guys who had a Picaro that they could zip around in and measure nitrous oxide at high resolution. And they did it all over the coast of South America here, or the eastern tropical South Pacific. And they detected a couple of hot spots, and this concentration is like 970 nanomolar. Remember I said equilibrium with the atmosphere was like 10. So this is a huge hot spot. And they calculated that emission from this spot could account for 20% of the annual flux of nitrous oxide from the ocean. And it's only one little spot. And they were only there once, well, twice. Didn't get quite that high there. So they hypothesized, and they showed that it probably was not being produced right there in the coast, but it was probably supplied by advection from where it was being produced deeper in the oxygen depleted water offshore. And before I show you a picture of that, I saw this and I went, I think I've seen that before. So this is a paper we had a long time ago in which we measured nitrous oxide, and that little hotspot is pretty much exactly that spot off Peru where they found 980, we found 200. So it's still a hot spot, and it means that it's reproducible in coastal regions that you have extreme nitrous oxide concentrations and close to the surface. This is a time series from Chile, same region, showing that episodically, just about every year, you have a high nitrous oxide production. And that's important to remember from this plot that the surface is, the maximum here is close enough to the surface that you could have diffusive flux. If you have a peak like that, you do have diffusion, but it's almost down at equilibrium there. So the way that it mainly is emitted to the atmosphere in this system is due to this upwelling phenomenon. And this is where the oxygen minimum zone is offshore, where nitrous oxide is being produced. Upwelling brings it to the surface where it can be emitted to the atmosphere directly. And this is poorly, both these processes and the proximity and the variability of the coastal system are very poorly represented. So the way that that works is we have nitrogen limited productivity. So therefore you get the highest production in the ocean where you have nitrogen being supplied from below by upwelling. That leads to a vertical flux of organic matter to depth, and its consumption consumes the oxygen as well. Anaerobic respiration then occurs, and that uses up the nitrate, and that leads to loss of fixed nitrogen via denitrification and anammox. And denitrification, but not anammox, contributes to the production of N2O and therefore to its eventual emission. That could lead to a little bit of increased global warming, which overall global warming is happening with or without nitrous oxide, which leads to increased stratification, therefore a lower nutrient supply. That leads back to more nitrogen limitation, but also to decreased oxygen supply due to the stratification issue. And even with all of that, it leads to more nitrogen loss, but I don't know if it leads to more or less nitrous oxide production or emission to the atmosphere. So my take-home lesson is that we need to know a lot more about the processes that can produce and consume nitrous oxide and constrain its emission to the atmosphere. So the first thing that was on my email this morning when I got here was a notice about a paper that had been published today by some friends of ours in the nitrous oxide world, of course, a harmonized nitrous oxide ocean observation network for the 21st century. I'm not the only person who thinks that we need to measure nitrous oxide a little bit better, especially in the coastal region. And so I think I'm going to leave you with that dissatisfying answer that we don't really know what's going to happen with nitrous oxide, but it's something to keep our eye on. So I'd like to thank the people who did a lot of the work that I've just shown you. These are the nitrogen babes. That's the back of the Nitrogen Babe t-shirt. And Al Duvall has been a very important colleague at University of Washington. I'd like to thank Sergey for making the mass spec run. And thank you all very much for being here.
Questions? Tim?
I'm trying to understand this amazingly interesting presentation. So the nitrous oxide is coming from very particular spots. That's partially because they have high upwelling, but also partially because they have high productivity and also low dissolved oxygen. But they have more low dissolved oxygen because of the higher productivity?
Yes.
Those three
Yes.
things.
Oh yeah. So he asked, I think you're asking, is the more denitrification, more nitrous oxide related to more productivity?
Yeah.
Yes, yes, absolutely. So I didn't really say this in the beginning, but the implication is that these are in upwelling regions, so there's a lot of nitrate in deep water. I showed you that picture of no nitrate in the surface water. It's below that. So where you have certain wind systems on the eastern boundary of oceans, you get upwelling of that water from a 50 to a few hundred meters. It's full of nitrate. That fuels the primary production. And then there's some vertical flux that sinks into that intermediate water and is consumed there, and that's what consumes the oxygen and leads to the process of denitrification. So the microbes.
And the upwelling also
Yeah.
is a path of getting into the
Right,
surface.
exactly. That's my idea.
Seems to be temperature limited, because you didn't show the same kind of hot spots in the cold.
No, well, there's no big upwelling up there, and the mixing that occurs where there's high productivity in the polar regions, is that what you're thinking?
Yeah.
That's well oxygenated. You get mixing in the winter to 700-800 meters up there, so you can have a slight oxygen minimum mainly, but you never get to anoxic conditions.
Rhizobia, which nitrogen they have an ion catalyst. Is iron sold in the nitrogen cycles in the ocean?
Oh, absolutely, yeah. So you said that rhizobia fixes nitrogen and requires iron, right? That was the statement, and therefore is iron important in the ocean? Yes, very important in nitrogen fixation, but almost all of the enzymes that are involved in these processes require iron. So iron can be limiting overall in the ocean, and there are places where it's the main thing that limits because it is a cofactor in so many enzymes. In the places that I was just talking about, it's mainly nitrogen because they're close enough to the coast. So that's where most of the iron comes from.
So I forgot about Jimmy's result that there is N2O production from nitrate in some of those incubations at relatively high oxygen concentrations. What are your thoughts about that, and were the rates important enough to affect these estimates for your basically understanding of N2O?
Yeah, well, there are a couple of peculiar things about that. So Danny says that he knows that there was production of nitrous oxide from nitrate as well as from nitrite, and that particularly for nitrate, it appears to occur where there's still oxygen. So that's partly what we were focused on last year on that cruise, was trying to figure out what's going on in the oxic layer of the ocean with nitrous oxide production and consumption, because we do detect it, and we detect the genes that are involved in encoding the enzymes for it. So I don't know if I believe that. I think we have to look at that a lot more. I kind of don't, but it's one of those things where, as you know, you've grown up as a microbiologist and oceanographer, you're like, no, that can't happen. But every time you think, no, that can't happen, then you're constraining your ability to figure out if it really is happening or not. So I think that's something we are working on.
So if nobody else has other questions, I'm just trying to make sure I really understand the story. So basically, the dominant factor that will decrease nitrous oxide over most of the ocean is increased stratification, which is going to be a result of global warming. But that would not have a significant effect usually in these upwelling areas, which break down the stratification. So that the extent the nitrous oxide is coming from your hotspots, that's really a different story.
So that the hotspots in the coastal regions are not in that model, are not appropriately represented in the model. And that might change things. But the question was if stratification warming is the main thing that causes a decrease in nitrous oxide production, then that won't affect the coastal regions or the upwelling. It's actually ultimately the decrease in primary production that is caused by the increase in stratification. So it's a double whammy, because it reduces the amount of organic matter that's actually delivered to the deeper water, and then it keeps it there with the stratification. So you get less emission, less mixing.
In your hotspot of upwelling.
Yeah, you think that might look quite different there. Yeah. So you might actually increase the yield and the production of nitrous oxide because of increased stratification, more denitrification, more sequestration, and then you could have venting to the atmosphere.
So maybe I missed it, but you said we don't really have enough information to make predictions. Basically, I think is your conclusion.
Well, I think we can make predictions, but they're highly uncertain. That doesn't ever stop anybody from making predictions.
What's a test or a measurement you think we could make that would improve our accuracy?
I think the most important measurement, and a really, really hard one, is what's going on in the coastal regions. And that's probably what I was hoping that observatory would include, the coastal regions, because one of the authors is a person who's measured, Rob Upstill-Goddard has measured nitrous oxide in a lot of interesting little coastal regions all over the world. And you find just a huge range of numbers that vary hugely spatially and temporally. And there's no way to incorporate that into models. And so I asked Rob at the meeting we were at not long ago, well, what would you do, you know? He gave a seminar on that, on the coastal nitrous oxide flux. And he never had a solution to that, because it's not easily generalizable. Right? I can't study it in my embayment and apply it to yours, or my river in India and apply it to yours in China. So I think a measurement system and understanding perhaps enough measurements to then figure out what determines where it is high would be most useful. Right? So you could use things other than direct measurement once you had a correlation or a principal components analysis or something that told you, machine learning, right, like we heard last week, to figure out what's going on in the coastal regions. I think that's the hugest unknown. That's mostly not in the models at all, or in those inventories.
Okay, let's thank Bess again.
Thank you.